Okay, so you can read the title there. There's two logos up the top, one of which I'm sure you're familiar with. The other one is the ISGC. It stands for International Sheep Genomics Consortium. So the work I'm going to talk to you about today, I've led within CSIRO, but it's also been conducted under the umbrella of this wider consortium. So by way of starting the talk, here's the sort of outline. I'll give you a couple of slides on this sheep consortium, the genomic tools that we've developed over the last couple of years. Then I hope to convince you that domestic animals are in fact a really good model for understanding evolutionary change. I'll do that through the prism of two experiments, the first of which is called the SNP50 HapMap experiment. I'll explain what that is. That's a completed experiment. Then the bulk of the talk, as the title suggested, is about genome sequencing to detect signatures of selection in sheep. And then the last couple of slides, I'll give you something about what's happening as we move forwards in the shape of the sheep genomes database. Okay, so the sheep consortium, this ISGC. So I'm the secretary. It's this kind of informal group of scientists. We talk to each other regularly, really with the main aim of reducing redundancy and coordinating what we do in the field of developing and applying genomic tools and resources related to the sheep genome. So each of these arrows is as I'll go through them, really they're tools and reagents that we've generated and we're putting to use. So the first and maybe the most influential of them is a SNP chip, so the SNP50 chip. We developed and launched at Plant and Animal Genome Conference in 2009 in partnership with Illumina. And I see Peter up the back, he'll remember this. Finally, we spent a lot of time identifying SNP, assaying and putting them on this technology platform, and then in partnership with Illumina made it available to the community. And what it allows you to do is collect genetic information from 50,000 loci spread through the sheep genome in an economically reasonable way, so you can go and genotype thousands of animals. So since then, people have been using it for things like genome-wide association, for understanding the relatedness within and between individuals within breeds and between breeds. And the sorts of things I'm going to talk to you today is about detecting parts of the genome that have been under accelerated change in response to selection to make animals look the way they look. So that was the SNP50 chip. If we fast forward now to last year, we've put together an HD chip. So we've moved from 50,000 now to some 600,000 assays on the platform. So that's a genetic loci that you can collect information from about every 5 kb all the way across the genome. And this is work primarily done by my colleague John McEwen and his colleagues inside Ag Research, and it would be remiss of me not to say that this was driven by my colleague upstairs on level 5, Brian, John and myself and others. Okay, so we've now got an HD chip. Really the big thing, and this is what's been driven by the team upstairs in CSIRO, is the reference assembly. So we've taken one male texel individual and one female texel individual and created the reference genome. So it's about 2.6 gigabase, which is pretty standard for a mammalian genome, and this schematic shows a whole lot of features through the different chromosomes. Take-home point here is we've got a really good handle on every base pair in the sheep genome, where they sit, but most importantly, it's been really nicely annotated. So we know where the protein coding genes are, we know the exon and intron structure of these protein coding genes through a data set that we generated over 800 gigabases. That's an enormous amount of RNA-seq data from 89 tissues from different time points. All got tipped in the top of an ensemble annotation pipeline to create these gene models, which are really at the heart of the annotation of our genome. So Brian is upstairs furiously about to submit all of this to a top-tier journal. Hopefully you'll see it come out sometime soon. So, a reference genome. Other tools we've developed include a parentage panel. This is under review at the moment in PLOS ONE. It basically describes what makes a good SNP for assigning parentage cheaply and effectively across breed. And the last one, which I'll get to at the end of the talk, is this sheep genomes database that is coming down the pipe in 2014. So they're the sort of tools and reagents that we've been busy creating and deploying on behalf of the community. Oh, and I should say that this consortium is an open platform. There's no membership. People come, people go. Everything that we make is public access. Everything that we do is for publication and for the good of the community. Anybody is welcome to join, and if you would like to send me an email and I'll stick you on the list and you can participate in the calls. Okay. So why might we use domestic animals to investigate aspects of evolutionary change? Well, the short answer is that the processes of domestication and selection to get us the domestic animals that we have running around fields today has been this magnificent experiment almost done on our behalf, and all we have to do is go in and look for what's been done to the genomes of these animals. So in the case of sheep, there's been a wild species, that's actually a North American bighorn, and the ancestral animal, or the progenitor of modern breeds, is in fact an Asiatic mouflon, but you get the point nonetheless. So there are two events in the population history of modern breeds which we need to consider for the rest of what I'm going to talk about. So if we go back in time, so if we go back something like 10 to 12,000 years ago, that's when man first started recruiting from the wild populations of sheep running around animals and controlling them. And it was really for access, stable access to meat. So through this process, you've domesticated these wild animals and you've reproductively isolated them and you've maintained them. Something then, in the hundreds of years ago then, we've siphoned these animals into discrete populations which we call breeds, and we've put selection pressure on them to make them specialised for some particular, for example, agricultural commodity. Think of dairy cows which have been specifically bred to produce milk. In the case of sheep, we've got carpet wool breeds, we've got dairy breeds, we've got meat breeds. Have a look at the size of this thing compared to the individual. We've got breeds which have been intentionally selected to turn off large amounts of very fine wool, etc. So there's been a phenotypic diversity across breeds for specialised commodities. But it's even more nuanced than that. We've bred animals to do well in particular environments, whether it be the tropical adaptation to high parasite loads. Conversely, there are basically desert sheep which are able to be quite productive on low nutrient plains, low water, etc., and wacky colour morphs and different flavour of horns and all sorts of things. So if we think more generally about domestic animals, this is why they're such a wonderful model to study evolutionary change. I like the example of dogs. So when I was at Cornell, I studied dogs. All of these different phenotypically quite divergent animals all came from the grey wolf. And so this process of domestication and then breed formation and selection in different ways has caused all of these different things. So we just have to go in and find the DNA that makes all of these different to a grey wolf. Same with cattle, with chickens, and even things like pigs. So this was Best Pig 2007 at the Royal Commonwealth Show, and you can see this character has been bred for lean and for growth, which is quite different to other selection objectives, for example, in this Chinese Meishan animal. What's the difference in the DNA of that one and that one that makes them A, different from each other, and different from their progenitor, the wild boar? So they're the sort of things that interest me. So to start answering that question, we can look for signatures in the genome of selection. And so this is what the schematic attempts to put across. So if we consider a before selection scenario over here, and we've got a population of animals, and each animal's got a pattern of diversity or genetic patterning along its chromosome. And in this character here, we're imagining that there's been some beneficial DNA mutation. It's founded on its chromosome that confers some phenotypic characteristic in that animal that animal handlers liked. Now, so in this example, European pig producers have liked white pigs since the 1500s. So in this example, that mutation might cause that animal to be white. And so producers have increased, so we've gone before to after selection now. And so in the population, you've got many more of the animals with the beneficial phenotype. Consequently, if you look at the chromosomes, you've increased the frequency of that beneficial mutation. And you've also carried along with it the pattern on each side of the chromosome. And so that's what I'll refer to as a signature of selection or a selection suite. And so those patterns that are caused by positive selection are reasonably easy to detect in DNA sequence. So you can use strategies like looking for runs of homozygosity, so where that selection is absolute and you've actually selected that mutation to fixation, so all of the animals are fixed in the genome for that position. You can look for runs of homozygosity. You can look for allele frequency differences. Obviously, you've mucked around with the allele frequency at that position and the neighbouring positions. And the third one I'll talk a bit about later in the talk is this idea of heterozygote deficiency. It's related to runs of homozygosity and to allele frequency differences. Basically, you've changed the allele frequency at and immediately surrounding the sequence variant that's under selection. So at the top I mentioned that there are two experiments I'll tell you about today. So I'll start with this first one called the HapMap or breed diversity experiment. So this was a large experiment that was concluded in 2012 where I wrote to a lot of people around the world and got some 3,400 DNA samples into the lab up on level 5. We made a repository. It was probably the richest collection of sheep domestic breeds anywhere, and we genotyped all of these animals, every one of them with the SNP50 chip. So we collected 50,000 genotypes for each of the 3,400 animals, but importantly the animals were from breeds all over the planet. So each one of these dots is a breed, not an individual animal, so we collected African ones, North and South American breeds, Asiatic breeds, importantly breeds from present-day Turkey and Iran, which is the domestication centre for sheep, so that's where the process of domestication started, and then breeds radiated outwards. And then lots of breeds through Mediterranean, Central and Northern Europe. What you see down here is now each one of these dots is an individual animal, and it's a cluster plot basically of the relatedness between animals. So animals that are closely related sit next to each other in the plot, and vice versa. Animals that are distantly related take different positions in the plot, and the colours, for most of them anyway, match the top. So these are Asiatic animals, African animals from the Middle East, and the green ones are all European. So we drew a lot of conclusions about the relatedness between animals and between breeds. Now, I won't talk more about the diversity stuff we did in this experiment. In the next couple of slides I'll really demonstrate that we can use this sort of data to identify signatures of selection. So I started with this character here. This is a Texel. It's a Scottish Texel. So we know quite a bit about these breeds. It's a muscle, basically it's a meat breed. But the thing in particular we know about it is it carries a mutation at the myostatin or GDF8 gene. And myostatin is a negative regulator of muscle growth. And this particular mutation in the gene basically releases the handbrake on muscularity, so that these animals have accelerated growth and this hypermuscled phenotype. Most sheep don't look like this. And we know that sheep producers have intentionally bred animals in the Texel to have a very high frequency of this G to A mutation in the gene. So we should be able to see a signature of selection in the Texel around that gene. So what I did was very simple. I just took the three populations of Texel that were in the experiment. There were German Texels, New Zealand Texels and Scottish Texels. I put all the genotypes from them in one pot, and then I put the other 79 breeds that weren't Texels in the other pot. Calculated for each SNP then the allele frequency in the Texels and the allele frequency average in the other 79 breeds. Expressed that difference between the two populations of animals using FST, and then plotted the FST for each SNP all the way across the the genome, and that's what you see. So that is SNP number 1 on chromosome 1, and that's SNP 49,000, which is sitting on the X, and all the SNP in between. And you can see this really strong signature of selection, where there's a big difference in allele frequency between the Texels and the non-Texels, and that sits right on top of the gene. So that proved to me, yep, absolutely, not only didn't we muck up all the labels on the tubes, which happens more often than it should, but the methodology to detect selection actually does work. So I'll only show you one other slide from the experiment, and it's, well, I'll step back on. the highest, well, the strongest signature of selection across the whole genome is this one that you can see on sheep chromosome 10. So that's chromosome 1, 2, 3, 4 and the interleaved colours going across. And here's this quite spectacular signature of selection on chromosome 10. So the metric on the Y here now isn't SNP by SNP FST. In this one, it's a bit more sophisticated, where you take the SNP alleles, you form them into haplotypes, and then you look for haplotype differentiation between populations. And the 10 populations I used, I haven't listed them, but they were all of the individuals in a breed were either all horned or all polled. And polled is the name for where the animal genetically doesn't have any horns. So it's not like you cut the horns off, it's just got a sequence variant that means that the animal's naturally hornless, right? And so here is this stunning signature of selection on chromosome 10, that if you dig into it and you look now just within the region and you ask, well, do you see it if you just compare horned breeds with horned breeds? That's the green line, you don't see it. Similarly, if you compare polled breeds to each other, you don't see it. It's only, you only reconstitute the signal where you compare polled and horned breeds. But about the time we found this, we did a GWAS, or genome-wide association study, in a breed that segregates polled and horned and found exactly the same gene. So this was very nice, in so much as not only could we see this startling signature of selection, but we could actually show what the target of selection was, and it's for the absence of horns. And there are lots of economically, really important sheep breeds from around the world, primarily of European origin, where people have bred them naturally to be polled. And if you want to chase this up, I'm proud to say we managed to get that published in a decent journal in 2012. Okay, so how am I going for time? So the second project then, and the title of the talk's about genome sequencing to detect signatures of selection. So this commenced in 2012, where we wanted to go from SNP 50 data to actually whole genome sequencing. Of course, this is very exciting because in your data set now are the actual sequence variants that directly underpin phenotypic variation. If you know where to find them, they're actually on your computer. The downside is we don't have money to go and sequence 3,400 animals, so that it has some consequences on the design and the analysis, how you do it. So the other rationale for going and sequencing genomes was to find the raw material for the HD chip that I mentioned at the top. So we've done that. We've sequenced some 75 animals, pulled out the SNP, and the first deliverable from the project is the HD chip. I won't talk more about that. Okay, so the coloured pictures here give some idea of the animals that we selected for sequencing. So you've seen that one before. This is exactly the same, but I've coloured in red those animals that we took forward into whole genome sequencing. And this is to convince you that we've selected animals through the whole parameter space, or the whole genetic diversity we've tried to capture in our selection of animals for sequencing. So we didn't just sequence lots and lots of animals from one breed, rather we've sequenced a small number of individuals per breed from a whole lot of wacky different breeds. Alright, there's a lot here. So sequencing and variant detection, this table at the top just shows the average number of megabases and the aligned megabases per animal. Suffice to say there was a lot of data. The important bit here is that we took the raw sequence from each animal, and I should say that Baylor College of Medicine in Houston did all of this sequencing, and they did an absolutely heroic job. Raw sequence from each animal, we mapped them against, each read against the reference genome version 3.1, so this is a Texel individual, and that creates then reference guided assemblies for each individual. We then submitted the .bam files that you get through that process to two variant detection approaches. One, Atlas SNP 2 is the name of the software that was done inside Baylor, and the other is SAMTools, which I'm sure some of you are familiar with. That was done by my colleagues at Department of Primary Industries in Victoria. And we only called a SNP if it was independently identified by those two pipelines, and they ended up in .vcf files for each animal. And so it's a bit of a different way to think about things that took me a while to get used to. So for each animal, you see variants where they're different from the reference genome. So this table down the bottom has individual animals per row, and so here's an African white dorper, that's its ID, and it had 9.1 million variants when its sequence was compared to the Texel reference. Of those 9.1 million, about 4.9 were heterozygous, and the remaining 4.2 million were alternate homozygotes compared to the Texel. Average down the bottom, about 9 million. So for the 68, well, we had some dropouts. We got rid of a couple of the genomes because we didn't have adequate depth of coverage to accurately call heterozygote positions, so we ended up with 68. Bit under 9 million SNP per individual, and you can see the other numbers. The other thing I should mention, although animals on average had about 9 million variants, across the 68 animals, we saw a total of 24 million variants. So we saw the same positions varied over and over in different animals. Oh yeah, annotation. So I mentioned at the top, ensemble gene models. So we compared our 24 million SNP against the gene models to ask the question, well, are they, which of these SNP are present in exons? Some, let me see, 178,000 of them in exons. Of those, about 64,000 changed the predicted amino acid sequence in a non-synonymous, and then we classified them based on the amino acid change. So all of that is available. So if we go back to this cartoon about selection, and now we think about, well, what analysis can we actually apply if we've got whole genome sequence? Runs of homozygosity, well, we can certainly do that, and I'll show you on the next slide what that looks like. Haven't applied FST simply because we don't have enough individuals per breed to represent the breed, so haven't done FST, but rather this idea of HDI heterozygote deficiency. Okay, so what you're looking at here is data from 4 individual sheep genomes, and I've plotted the 250 megabase of sheep chromosome 2, and on the X, I've binned SNP into 20 kb physical chunks, and then for each bin, plotted the percent of SNP in that window that were heterozygous. And you can see it wobbles along, and then you get to regions of the genome where the percent heterozygosity plummets to 0 because they're homozygous. There is no variability on their chromosome. And you can go through and you can plot where these regions are in each animal. And this individual happens to be a Texel, and here's our friend GDF8, sits in the middle of this run of homozygosity. And that's one approach. I'm not going to talk more about it today. My colleague Cuttell is plotting where these occur and where they appear to occur in the genome more often than you would expect by chance, because that can occur either because that animal has GDF8 and there's selection, or because the animal has received 2 copies of that haplotype through its pedigree just by chance, and it's got a high proportion of co-ancestry. So you can get these not because of selection, but just because of drift and pedigree. So what I'll talk about for the rest is this metric HDI, or heterozygote deficiency. Sounds complicated, it's not. What I did was I tested each SNP for Hardy-Weinberg equilibrium. So if you think back into the, well, for me anyway, it's a number of years ago that I sat in lecture theatre and heard about Hardy-Weinberg equilibrium. If you think of a naturally mating random population and you look at a biallelic SNP, Hardy-Weinberg says you should have a quarter of one homozygote class, you should have a quarter of the other homozygote class, and then 50% of the heterozygote class. So you've got three classes, should be 25, 50 and 25, right? Where you see a significant deviation from those ratios, you transgress Hardy-Weinberg, and that's what I'm testing here. So I simply apply the Hardy-Weinberg test to the genotype classes derived from the 68 animals at every SNP. I then bin the SNP into 20 kb windows, and what I'm going to tell you, or what I'm going to report, is the proportion of SNP per window, 0 to 1, that either transgressed Hardy-Weinberg to 0.01, so had skewed allele frequencies, or 0.001, desperately skewed allele frequencies, generally where one of the 3 genotypic classes isn't present. So if you split your genome into 20 kb windows, you get about 250,000 of them, and here's the distribution of the number of SNP per window. So on average about 150 SNP per window, and I chucked out the ones where there was less than 20 because that enriches for false positives, reasons I won't go into. So here's what it looks like across the genome. So you've got chromosome 1, 2, etc. across to chromosome 26 and then the X. And here's this proportion from 0 to 1. So just to remind you again, that's a 20 kb window where nearly 100% of the snip in the window, and I average there's about 150 snip per window, nearly every snip in the window has got a very badly skewed allele frequency, and so transgresses Hardy-Weinberg. And you can see that absolutely there's some structure here to the data. So what are the genes under these peaks is immediately what I was interested in. So I ranked regions based on the metric, and hang a minute. Right, no, I didn't. The first thing I did then was to ask, well, can we see things in the data that we would expect to see? So here's our friend Pole Horn again, and the RXFP2 gene. So this shows the approximately 90 kb on sheep chromosome 10. That's sort of been chopped off the top there. There's a little asterisk there at megabase position about 30, which is blown up here. So that's the snip per window, and here's the two metrics either at 0.01 or 0.01, and immediately sitting under there is the gene. So you could re-find, using this new approach, what we already knew to be under selection in sheep. So that's encouraging. Our old friend again, GDF8 and the Texel. And in fact, we would expect not to find this. And the reason is that this particular selection sweep is private to the Texel. It's only been selected in Texel. And given that there were only 2 Texels in a set of nearly 70 animals, we wouldn't expect it would slant the Hardy-Weinberg result enough to actually detect it. And you don't see it. So it's good to know when you design an experiment and you're applying a metric, well, what would you expect and not expect to find? And in this case, we wouldn't expect to find selection sweeps that are only present in 1 or 2 breeds. Rather, we would see selection which is common to many breeds and therefore is probably the sorts of selection that has occurred at domestication and prior to the radiation of most sheep breeds. So we're looking for old selection sweeps, not recent and breed-specific selection sweeps. I reckon I've missed a slide. Okay, so to do this meaningfully, you have to set a threshold, and you have to set that threshold such that anything that jumps over the top, you say that's a candidate for selection. Anything that doesn't, you say probably hasn't. So I decided we'd set it at a really, really highly stringent level, so greater than 10 standard deviations away from the mean. So this is the distribution of the metric, and you almost can't see it. So basically we declared 321 of the windows as significant out of the 250,000. So the absolute right end of the distribution of the metric. So those 321 windows define 137 regions. Those 137 regions, 97 of them had genes for a total of 366 genes. So I've got this whopping table that starts at region 1, goes down to region 137, and has got what genes are in there. So we're getting up to the point where the experiment's up to. So if you rank them based on the strength of that selection as measured with HDI, this is what you see from the most strong downwards. The ones I've coloured in red are ones that we've got pretty strong a priori information to say that they're under selection. So there's RXFP2, so that's the strongest in the experiment. So I went through and fished out those genes and those regions for which we've got really quite good evidence of selection relating to pigmentation, wool, body conformation and production traits. I won't go through them all. For those of you that have got an interest in genes that are involved in production traits and things, if you see anything on the list that lights your eyes up that's not in red, come and tell me afterwards. It's very unsurprising that we see strong selection around pigmentation genes, not just in sheep, but even in lines of mice and cats and dogs and cattle and pigs. Coat colour and coat pigmentation, breeders have liked their animals to breed true to type for a long time. If you've got a brown animal and you cross it with a brown animal, you'd like to get a brown animal. So pigmentation genes, EDAR, NF1, EDM3, this is the agouti locus, which we already knew causes pigmentation changes in sheep. Interesting ones concerning wool pigmentation, vitamin D receptor, a lot of literature around its impact on follicle, primary and secondary follicle development. Body conformation, I'm interested in this one, Link28B. It's known in GWAS from humans to control the age or the onset of puberty. And so in domestic animals, that's one of the first things we did, control the reproductive cycle. And for most species, we've bred animals to reproduce earlier. So Lin28B being under selection is interesting, and a whole lot here for muscle traits, milk traits, and some disease genes. That says the same thing in a different way. Okay, so in a couple of minutes I've got left, we'll dig into some results just specific to one chromosome. So this is sheep chromosome 3. So in the blue up the top, you've got the SNP per 20 kb window. You've got the metric at 0.01, and then the metric again at 0.001, and you can see it sort of squashes down the noise. And the 3 peaks that I'll talk about here, that one sits right on top of the EDAR gene, that one the HOX genes, and that one MSRB3. So MSRB3 is known in pigs to control of all things ear morphology. So the difference between pricked ears and big floppy ears has been sheeted home to MSRB3. But I'll talk about the other two. So the first one I'm going to talk about is this signature here, which is at about megabase 130. So I mean, this is just like turning on the light. Just here, there's this massive selection signature, and the next slide just blows up between these two lines, and you can see it starts at the HOXC4 gene and it finishes at the end of the cluster here. So we know a lot about the HOX genes. It's 4 clusters, A through D. This is C. I went and had a look, and you can see evidence for selection around the HOXA gene cluster. Didn't reach significance, but no signal at all around B or D. So this is a really nice review of the consequence of the regulation of gene expression off these HOX gene clusters. And what they are basically is a GPS system effectively for guiding body shape in the 3 dimensions. So the transcription off the A cluster dictates the distal proximal body shape, and then off the C cluster, the anterior posterior shape. And both of those axes, it would be reasonable to say, has been under selection since domestication. In pigs, for example, we know there's been, domestic pigs are shorter and they're longer, so there's more vertebrate in a domestic pig than a wild boar. I think there's 2 more averages. Same with sheep. Most of our domestic breeds of sheep are considerably shorter than bighorn, thinhorn and Asiatic mouflon. So it's interesting that there's this really strong signature of selection around the HOXC cluster. Now if I take you back here, so HOXC12 and 13 sit under the peak of the peak, if you like. And they're of particular interest. There's a lot of literature about their coordinated expression, where they're expressed in wool development. So here's HOXC13 and 12 in the developing primary hair follicle. And what these 2 genes do, or the expression of these 2 genes, is they control the process of anagen, which is the wool growth cycle. And if you think of a merino, this animal has been intentionally bred to have all this wonderful, luxurious long wool, and they've got this extremely extended anagen period of growth cycle, hair growth. So it's now for us a really strong and interesting couple of genes to relate to wool biology. Okay, this last couple of minutes, the other one I'll talk about is this one here, EDAR. So this is what it looks like. Here's the peak of the signature of selection, and there's 3 genes under there's EDAR and these other 2. So this variant in EDAR in humans has been shown to be under selection and responsible for straight hair in populations of East Asiatics. And further, in this paper by Pardasabetti and colleagues, they actually went to the trouble of engineering this specific mutation into a mouse and reconstituting the straight versus wavy hair phenotype. Partly, I don't think that they are particularly interested in hair biology, but they wanted to prove that the paradigm that I've been telling you about today, signatures of selection, actually does find things that have a phenotypic consequence. So that counts of interest. Anyway, so we see in sheep either highly selected or under positive selection, or a candidate for it at least. And so it stands to reason, given wool phenotypes are under selection in sheep. So I mentioned there were 3 genes in the region. Here are the other 2. So each of these is one of the 68 genomes, and you can see there's an accumulation of homozygotes and a loss of diversity on top of the EDAR gene that's not as prevalent on the other 2, suggesting that that is the gene driving the signal. And looking in the annotated SNP, absolutely you can see SNP through to exons, one of which is non-synonymous. Now I don't have any additional proof it's circumstantial whether that's got anything to do with hair biology and it's been under selection, but nonetheless there it is. Okay, so conclusions in the last minute or so. Absolutely, with these data sets we can confidently identify regions of the genome and the genes inside them that appear to be under recent positive selection. We can use SNP50 data to do it. More recently we can use sequence. But what next? Like these studies, as I said, you can identify regions and genes that appear to have experienced positive selection. It's much harder to actually refine them down to the sequence variant that is the one that causes the phenotypic variant. So to start getting at that, next thing to do is to overlap the SNP annotation onto the suite of regions and come up with candidates. And even this is simplistic because it assumes that all phenotypic variation is the result of protein coding change, which we know it isn't. There's lots of other things in the regulatory regions of genes that have phenotypic consequence, but this is what we can get a handle on in the, at least to start with. So if we do our job right, we'll be able to resolve at least some of these to candidate causal sequence variants. And then in the last 2 slides I'll tell you about the sheep genomes database, which is related to all this. So the concept here is inside CSIRO, in this consortium I've just told you about, inside AgResearch, USDA, and well, there's labs even in Brisbane that are creating data sets using sheep as a biomedical model. It would be great if we had a single clearinghouse for all of these genome sequences so we could stick them in a standardised format and then make them available to everyone. And so we convinced USDA to give us some money to do that, and that's what the sheep genomes database is going to be. So sitting inside this database will of course have the reference genome, people with eligible data, and that eligibility criteria will be about depth of coverage and a couple of other things. Can submit their BAM files in a unified way. We'll call variants, whether they be SNP indels or CMV, and using the genome, annotate them where we can, and then make available to anybody that wants them the data, which should be useful for candidate gene studies. If you've got a gene of interest, you can go in there, pull out the SNP and figure out which ones might be of use. Loss of function mutations, and I put in red here the things that I'll be attempting to achieve. So that's a CMV, a copy number variant atlas, using the variation in depth of coverage for all of these animals. Imputation for genomic prediction is going to be important, not my interest, but colleagues, I'm sure will put it to good use, and the evolutionary studies that I've been talking about today. So the first 68 genomes I told you about today have gone in, and as part of this USDA grant, we've got money for another 200 exomes that will generate during 2014 and put in. And I would say by the end of this calendar year, we'll be up to 250 or 300 genomes beyond those there. Okay, so the most important slide, I think, is the acknowledgements. So my colleague Brian, Seen, Juka, Yutao, and Jan Yu inside CSIRO. At times, I would say we communicate, if not every day, every second day, with our colleagues in AgResearch, John, Rudy, and Shannon, and their colleagues. Mike Heaton at USDA did a QC job that pulled this whole experiment out of the fire and got it back on track, for which I'll be eternally grateful. Noelle and Tracy at Utah State, Ben and Hans at Big DPI, Kim Worley and her co-conspirator Richard Bay. Kim and her team at Baylor just did a magnificent and continue to do a magnificent job, and Marty and Gordon McCart at the University of Montana. And with that, I will stop.
Hi everyone. Yeah, Stephen's right. The nomenclature in parasitology is very confusing. It's confusing even for us. They're trying to become more consistent. So today I'm just trying to remember that I should call it beef tapeworm for you guys, but the scientific name is Taenia saginata. I'm going to talk today about some work that I did in 2012, a postdoctoral fellowship that was funded by Meat and Livestock Australia while I was at the University of Queensland at the School of Vet Science. Other people who worked on this were Helen Owen, who's a veterinary pathologist at UQ, Ella Leigh, who you will know well, and also Rebecca Traub, who's a veterinary parasitologist, originally based at UQ but now at the University of Melbourne. I'm going to talk about the development of a real-time PCR that we developed in order to enhance the classification of the infectious cysts of beef tapeworm at meat inspection. Don't panic, I'm not going to talk too much about the technical issues of the real-time PCR. I've kept it clean and simple for you. But I'm going to spend some of the time talking about how inspection is currently done in Australia, the reforms that are taking place at the moment, and how we can use molecular diagnostic tools to help improve food safety outcomes for consumers. So the beef tapeworm is a zoonotic parasite of humans and cattle. In humans, it causes a disease, taeniasis, which occurs when beef tapeworm is harboured by the human in the gut, and segments and eggs are shed in the feces. Cattle are the intermediate hosts, and they become infected when they eat feed or pasture that's been contaminated with human feces, and the eggs or the segments will develop into larvae and then cyst in the muscle tissue. They look like this, and here's where they can be called cysticercosis, cysticerci, metacestodes. I'll just point out that this scolex here, or the protoscolex, is a diagnostic feature for Taenia saginata in muscle tissue. And the cycle is complete when humans eat undercooked or raw beef that's infected with viable cysts. In Australia, we have Taenia saginata. It's a global parasite, and it's found in all states around the country, but it's at a very low prevalence. So the last time they did a survey, it was estimated to be as low as 1 in 500,000 head. When we do have disease incidences or outbreaks in Australia, it's typically related to poor hygiene practices by humans. A really old example, but a really good one, was at the Werribee Sewage Farm down in Victoria. This is a large state-run farm where they used to use, or they deal with the majority of Melbourne's wastewater, and they used to irrigate pasture for 15,000 cattle with the sewage. As a result, up to 33% of the cattle were found to be infected, and luckily they don't do this anymore, they use recycled water. We also had an outbreak in 2010 in northern New South Wales. That was the result of contaminated copra that was imported from Papua New Guinea. The copra can be used as a feed supplement. It's a dried kernel of coconut, and it was contaminated with human feces. So beef tapeworm is one of these foodborne diseases that actually has quite a low public health concern, but the main impact is through its economic impacts to the beef industry. So an adult tapeworm in a human has quite minor health implications, things like anemia, constipation, and abdominal pain. That's a pretty low public health concern compared to something like Taenia solium, the pork tapeworm, which we don't have in Australia, but that can cause neurocysticercosis, which can cause seizures, blindness, and even death. So although it's not a major public health concern, it is still regarded as a major food safety issue in both domestic and export markets. And the costs are mainly borne through the intensive inspection approach that we have. So every carcass or every animal that's slaughtered is inspected by a person to make sure that there are no cysts. And there's also significant losses through condemned meat and offal. In Australia, meat inspection is regulated by the Australian Meat Standard. And in regards to beef tapeworm management, domestic and export beef is required to be visually inspected by incision. So this means that the meat inspector or the on-plant veterinarian will make a number of cuts into usually predilection sites such as the heart or the masseter muscle in order to try and find the cysts. The disposition includes heavily infected carcasses need to be removed from the food chain, but the standard does allow trimming of cysts plus freezing to render any of the cysts non-viable in light infestations. So post-mortem inspections pretty much been the cornerstone of consumer protection in the red meat industry for well over 100 years, but it has been questioned, in particular the beef tapeworm management, because visual inspection tends to lack accuracy and precision. It's considered inefficient because the meat inspector needs to balance the number of cuts he makes with not causing as detrimental an impact on the meat quality. So he needs to make enough cuts to detect those low-level infestations, but the more cuts he makes, the lower the product quality becomes, and there's also an increased risk of microbial contamination, and that can cause a far more serious food safety issue than beef tapeworm. More importantly for this work is the poor positive predictive value of visual inspection and incision. So there are a number of other organisms or parasites that cause macroscopic, morphologically similar cysts in beef muscle that can be confused with beef tapeworm cysts. And a lot of them are non-zoonotic, so they don't have any public health concern at all. That's bad because if a meat inspector has a suspicion of beef tapeworm, he'll withhold that carcass from the line, which reduces in further economic losses. The traditional laboratory method is histopathology, and the degeneration of cysts or lesions can particularly affect this method. So cysts generally remain viable for 3 to 6 months in the bovine host, but after degeneration starts occurring and the immune system's kicking in, you lose that diagnostic feature of the protostolics, which I showed you at the start, and the histopathology is pretty much useless. Sorry, pathologists. And just to illustrate this point, these two images are of beef tapeworm metacestodes, so what a meat inspector might see after making an incision. And other things that are commonly misidentified include this guy, cysticercosis. This is a common protozoan parasite that's found in beef muscle. Other things like Echinococcus granulosus, Tinea hydatogena are commonly found in beef muscle, but they don't pose a threat to human health when in beef. And I included this image here because during our submission process, even neoplasms or sarcomas were submitted, which shows that even non-infectious lesions can be confused with beef tapeworm cysts. And that leads to, again, unnecessary withholding of taxes. So I've talked about how visual inspection takes place in Australia at the moment. But there are proposed changes to this type of approach. There are global meat inspection reforms going on, and Australia is also moving towards that, towards a more risk-based approach. This requires developing risk profiles for beef herds. Then you are able to apply greater scrutiny to high-risk herds compared to low-risk herds, and it's anticipated that this would result in significant cost savings to the beef industry by reducing the amount of inspection in low-risk animals. For example, high-risk herds in the cooler, wetter areas of Australia, we find that Tinea saginata prevalence is higher than in areas like the hotter, drier parts of the country. Also, a high-risk herd, animals coming from a property that's maybe had an outbreak before, or the source of infection has not been identified, they can be flagged on their way to slaughter to be inspected. So in order to contribute to this overall improved management program that's been proposed, there is a requirement for a better diagnostic tool in order to reduce the number of those false identifications at visual inspection. And so that's when MLA put out a tender to develop a fast, specific diagnostic molecular assay, which we were successful in, and we decided to pursue a real-time pro-based PCR, which offers all the usual advantages of PCR of high sensitivity and specificity, and in contrast to already published conventional PCRs, offers an additional advantage of reduced risk of contamination by being a closed system, because there's no gels running obviously, and also fast turnaround times. Fast turnaround times are desirable because if a carcass is withheld on suspicion of beef tapeworm, it can re-enter the processing chain if you are able to furnish a negative result fast enough. Now it's a bit of the boring bit, sorry. So here's some of the validation methodology. We designed actually a duplex reaction that simultaneously amplified the beef tapeworm specific and target, and also an internal control target. If anyone's interested, it was mitochondrial COX-1 for both. We selected bovine DNA as the internal control because inevitably bovine DNA is co-extracted during the DNA extraction process of the cyst. And an internal control is necessary in a diagnostic laboratory because it demonstrates the presence of amplifiable DNA and the absence of PCR inhibition. We did the normal validation studies, estimations of analytical and diagnostic sensitivity and specificity, and also reliability assessments. So these are studies of repeatability and reproducibility in true positive and true negative lesions across 2 laboratories. I mention this because it's an important aspect of validation that tends to get overlooked. It's very important to make sure that a diagnostic test is transferable across laboratories, that it works just as well in another laboratory that it needs to be applied in as the one it was designed in with that operator. We looked at the applicability of the real-time PCR in suspect lesions submitted from Australian abattoirs. For this, we went directly to export abattoirs because at these ones, there's a government-employed on-plant veterinarian as opposed to a trained meat inspector at a domestic slaughterhouse. And because of their extra veterinary training, they should be better able to differentiate parasitic lesions from other pathologies. And we looked at the agreement of the real-time PCR assay with results of the traditional laboratory method of histopathology and also a previously published nested PCR that we used as the gold standard. When samples were submitted to UQ, they underwent a morphological classification and identification using the laboratory methods. So in terms of morphological classification, cysts were put into one of these 4 categories. Viable cysts is cyst-shaped and they're fluid-filled, and when you cut them open, you should be able to see the protoscolics under a stereodissecting microscope. When the bovine immune system kicks in and the degeneration process starts, the cavity tends to fill with this cheesy caseous material. At this point, they're not infectious to humans anymore. When they're degenerated, they're no longer cyst-shaped, they become quite hard, and all the way down to highly degenerated, when they basically just become non-specific multifocal granulomas. Just to show you, we did our homework. The assay was tested with DNA of host and non-target parasites, so other closely related tapeworms, also protozoan and bacterial parasites of tissue, as well as host. And you can see that it was specific. This amplification signal corresponds to beef tapeworm. And it met good efficiency criteria and was sensitive to the level of 220 copies of beef tapeworm, the target, mitochondrial cytochrome oxidase C. I put in this slide, it's a nice simple slide just to show you the ease of interpretation of this assay and how that's different to conventional PCR. So as you would know, you need to run your PCR product alongside a size marker matched up, and that can be quite subjective sometimes and lead to longer turnaround times if you need to investigate a product that doesn't match up properly, maybe running it longer or retesting it completely. So this is the beef tapeworm standard, 1 signal, internal control standard, 1 signal, and in a true positive lesion we get 2 signals. So it's very clear result and easy in the diagnostic lab to interpret. I guess this is the meat and bones of the talk. This shows the performance of the 3 different laboratory methods in their degree of ability to positively identify cysts of varying viability, viable, calcified, degenerated, and highly degenerated. You can see that histopathology was a poor performer and was only able to positively identify viable cysts. Compare this to the PCRs. The nested PCR was able to detect as low as highly degenerated cysts compared to the real-time PCR, which could only detect the degenerated cysts. This is largely because of the increased number of cycles, 65 compared to 40, but the real-time PCR outperformed this PCR when only 40 cycles were used. This nested PCR comes at a cost of increased turnaround time, cost in labour, cost in reagents, and you can see that's reflected here. The real-time PCR only takes 6 hours to get a result compared to 10, and it's actually cheaper because of the reduced labour component. So just to summarise that, it doesn't detect the most highly degenerated examples, but it does perform quite well, and it offers fast turnaround times and lower cost. And I'm probably close to my time limit, so I'm just going to finish off with some nice warm and fuzzy conclusions and self-congratulation. But basically, our test enhances the classification of suspect lesions, and it provides savings for abattoirs as carcasses can be returned to the processing chain if they're found to be negative, and it enhances our reputation for excellence in food safety and quality control. And I'd like to thank Meat and Livestock Australia, and in particular Dave Jordan and Baden Pearce. These people helped with the sample collection, and these people helped with the research and data analysis, And I've left off an important person, Leigh, who's also at the vet school at UQ, who helped with some of the sensitivity and specificity data. That's it. Thank you.